So our last speaker for today is Dr. Jacek Lubelski, Chief Technology Officer at Nanocell Therapeutics, a biopharmaceutical leader specializing in gene and cell therapy and viral vector production. With over a decade of experience, Jacek has advanced RAAV therapeutics and now oversees R&D activities at Nanocell Therapeutics. He'll be discussing novel non-viral DNA-based gene therapy vector for CAR-T engineering in vivo. Please.
Thank you very much. And let me start by thanking the organizers for having us here today and for the ability to discuss some of our data. I'm going to talk about targeted lipid nanoparticle, but we will switch gears a little bit towards preclinical. So I'll mostly talk about what can we do with those vectors, and I'll show you what we at Nanocell have developed. But before we get there, let me start with CAR-T. And by the way, thank you for staying until the end. Are you familiar with the CAR-T? CAR-T has been a tremendous success. This timeline just shows a number of different scientific discoveries that actually were put together or contributed to actually developing a product, developing a drug, which first went into patients in 2011 and was accepted by FDA in 2017. And we now have, I don't know, I stopped counting, 5, 6 different ex vivo CAR-T products. It's tremendously successful with overall response rates topping 80, 90% in some of the indications in relapsed patients. However, it's not without problems. This is how CAR-T ex vivo are made. So you need cells from a patient. They need to be modified outside of the body, usually with lentiviruses, and then permanently, and then they come back to the patients as a personalized medicine. So you can imagine there are a number of disadvantages of that therapy, even though it's so effective. And we could go through all of them, the complex manufacturing, the cost, and so on, but what it really stems down to is the access of patients. These therapies, and let me quote just one review that says that in DLBCL alone in the United States, only 20% of relapsed patients that are eligible for that kind of therapy actually gets that therapy. In Europe, it's very variable throughout the countries. I've been reading several reviews about whether these therapies, these advanced therapies, cellular therapies are available, and it really differs from country to country. So there is a lot to improve there, and there is a solution that is being developed, and actually it's already in a clinic. And I borrowed this table from Asher Mulder from this review to illustrate how many of those different modalities which can be grouped into lentiviruses and LNP are being developed. And it's not a cell therapy anymore. This actually is a gene therapy. Because these vectors are used in order to modify the cells, the CAR-T cells, but not outside of the body, but actually inside of the body. And therefore, they have potential to avoid a lot of those complexities related to personalized manufacturing. And also, they have potential, which is important, to decrease the costs. And I also put the quote here from that publication. So they say, the beauty of what we do, and they say we have that on the wall, is that it's faster, better, and cheaper. And I think those three will also be important in discrimination between those different products. Which of them would be faster, which of them would be cheaper, and which of them would be better. So what we are developing at Nanocell is actually non-viral targeted LNP that contains DNA and RNA. And why do we do that? We do that in order to kind of harvest the best of both worlds. So you can see that there is a lot of lentiviruses that are being developed. Some of them are in the clinical trials at the moment. And this is an incremental development because Lantia used ex vivo. So the next step would be obviously to try to actually modify those cells in vivo. They take advantage of permanent modification, an advantage of the proliferative potential of the T cells, and then growing of the drug with it. The non-viral RNAs, they're transient. So they deliver the LNPs with RNA. They'll deliver RNA, and then you need to redose. But they have a lot of advantages from a manufacturing point of view, from a cost point of view, from modularity and throughput point of view. So that's why we would like to decide to try to combine the two and develop a vector that is actually integrative, but it's still non-viral. It's LNP. So how does it look? It's a targeted LNP, so it contains a protein outside that has two domains that are used for activation and targeting. So targeting is important because we now go from ex vivo to in vivo, and we need to be specific in order to avoid certain potential safety risks. It also needs to activate these cells because the cells that we encounter will be resting. So we need to activate them in order to transfect them, actually. And for this, we have, when we are inside the cell with this LNP scaffold, we deliver two types of nucleic acids, DNA and RNA, and it's the transposase transposon pair to integrate this mini circle that contains the CAR, the Chimeric Antigen Receptor, into the chromosome of a T cell, and they take advantage of the proliferative potential of the T cells and then to grow the effect. And what I would like to do today, and I promise I'll keep to the time, I know I'm last. So I would like to talk about the preclinical package, how we're actually developing the evidence for that vector that is effective and it's safe. And I'll talk about some data on the pharmacodynamics, so essentially an efficacy of the product, what the product is doing to in this case, model rodents, humanized mice. And then I'll go to talk about a little bit about pharmacokinetics from a perspective of biodistribution. So how the product is distributing, where we can track not only the entire particle, but also, importantly, components of that particle and its functional delivery. So we made a series of vectors that contains different CARs, CD19, 22, and the dual CAR, which are our lead clinical candidates, also to demonstrate that we can achieve this efficacy, not with one vector, but with multiple. Also, we used two different rodent models, the NSG, so the immunodeficient mice, that are reconstituted either with PBMCs, that gives these mice sort of human immune system, or parts thereof, and this type of model is used for very rapid study because these models, those mice, they get to VHD very quickly. These ones, which are HUCD34+, that are reconstituted with cord blood, they have a little bit more physiological, let's say, different cell lineages, human cell lineages, and they also have delayed onset of GVHD, which is important for us because what we do is not transient generation of CAR-T in vivo, but it is permanent, so it's long-lasting. And with this model, we can actually demonstrate that upon single administration of very low doses of that type of vehicle, we have a generation of CAR-T that lasts around 140 days in those animals. And this is what I would like to discuss with you today. So I will show a series of experiments which looks very similar to this. So we have a mice, then we have a non-6 tumor cells that contains luciferase. This is for in vivo tracking of actually growth of the tumor mass. Then we have sort of immunoreconstitution either with PBMCs or with the cord blots, depending on the model. And then we come back with a tLNP that has different cargos, GFP, or the CAR, different CARs. During the experiment, excuse me, we monitor the expression of the CAR in T cells. And then also we look at the tumor, how the tumor is developing at the end. We sacrifice those animals and we'll look for either signs of expression of the CAR delivered by this vehicle, or later on I'll show you also some biodistribution data pertaining to DNA and RNA. So let's start with the CD19 first. So CD19 is a typical a model, let's say. And what we've done here, we used the animals that received PBS as a control. You can see that this red dots on the mice, they signify the tumor growth at the day 29. All those animals, they need to be sacrificed because they reach human endpoint. Whereas if you add 40 micrograms per kilogram, and I will keep underlining those here, which is important also for its safety and biodistribution, you have a full control of the tumor. If you go with even less, 4 microgram per kilogram, you have a partial response. And in this little plot, you see that for these protection of those animals, these cells are responsible, which here are plotted as a CAR T cells per microliter of blood. But if you would look at the percentages, there will be around 50%, 40% of all circulating T cells in these animals will actually have CAR. And this is what you see here, but in this case with our lead clinical candidate, which is called NCTX-01, you can see that in blood with the NCTX we have around 60% at the day 20 after single injection. And we also have CD22 here, which is a little bit less. And then if you look at the organs after the sacrifice, again, you see that in those T-cell-rich organs where T-cells are residing, also we have a lot of CAR expression, showing essentially that we can effectively generate CAR T, and these CAR Ts, as you see over here as well, they are functional. So here, instead of PBS, we use GFP with the same results, so these animals are not protected, obviously, and at the moment they start dying. With the NCTX-01, this CAR is very effective, and we have already at day 20, we have full protection of those animals and no visible signs of generation of NAM6. And then with CD22, it works as well, but to a lesser extent. Then we move to a different model. And this model is important because, as I mentioned before, what we would like to demonstrate with this model is 2 things. First, can we generate these CAR Ts that are long-lasting? And that's the promise of the integration technologies. They need to last much longer than the actual mRNA. And first, you see over here that with NCTX-01, we again generate a lot of CAR T in the blood. These are B cells. The B cells, they contain CD19, which is a target. Based on this, they are being killed. And you have B-cell aplasia. This is the most classical setup for this experiment, let's say. But what we've done, in order to demonstrate the longevity, we also add tumor. And with this tumor, we generated, again, a response over here that you see in the control animals, day 21, we need to sacrifice them. In the group of NCTX-01, you have majority of those animals, they survive for a very long time. There are some that did not respond. And that was interesting for us. And we start to look for the reason why actually they not responding. And it became very evident that when we looked at the day -1, so before injection, when we look at the amount of T cells that they have, it correlated with the response. So if you don't have T cells, you can't generate CAR T. So in a sense, it supports the mechanism of action. This model is known from being variable, and some of these animals, when they sold, they just have human cells, but these are not T cells, and this is what you can see over here. Now, next, what we wanted to do is to first show this in time. Can we have the T cells? Will they react to the target? Will they go after? What happens when the tumor is actually removed? And this is what you see on this plot. So the red line is the tumor load, which you see over here. And we injected those animals with the tLNP, and they immediately shoot up a production of the CAR T. And this production will go down. And you see the tumor here is removed completely. On those previous pictures, you can see them. So this nicely expands. That's what it should do. And then it goes down. And that's, again, what we have expected. Now, in order to demonstrate that this is long-lasting, we re-challenge those animals. And this is the line. So we give them an extra NAM6. We give them a relapse. We give them tumor. And you can see that immediately the tumor load go up. And then we did not inject the tLNP here anymore. Those cells were there, and they shoot up again. And the moment that they start killing, controlling, fighting with the tumor, they'll go down. Few words, I'm looking at the clock, but I think I still have quite a good 15 minutes, right? So then let's move to the blood, because this is also important or interesting how those tLNPs, they actually behave in either isolated T cells, and this is what you see on the left side, or directly in the blood. And let me explain the experiment. So this one, we just took T cells, we isolated them, we give them the tLNP, and you can see that we achieved a very high signal from the DNA. This is important. It's not RNA. It's really coming from the DNA. And then the signal is when we culture them for a prolonged period of time, until 20 days, you see that the signal is stabilizing, not all of the signals incorporated, but you can see a population that is stable. And this population, as I will show on the next slide, we used for the killing assays, demonstrating that actually it kills the cells with the antigen. Even more interesting, we look at the blood of the healthy volunteers, and the experiment was very simple. We just add the tLNP that we made, and we add anticoagulant. That's it. We mix it, and we leave it. After 4 days, we see CAR appearing. And interestingly, in this blood, there are B cells, which are the targets. So they start to serve for a CAR, and they expand the CAR. So you see that the CAR T are growing here and growing, and you can see that the number of them is actually start dominating the pool. And exactly, this is, again, exactly what you would expect. Okay. So what I wanted to say with this one, and I will not have the time to show you all the data, but we also tested a number of patients' material. Patients' T cells will be different. There are different indications. We tested DLBCL, we tested ALL, we tested also some neuroblastoma patients. All of this with the same results. So we take the leukapheresis material of that patient, we give the tLNP, and we can generate CAR T. Then we have a dual CAR. In this dual CAR, we have 2 domains that are targeting CD22 and CD19. And we wanted to demonstrate with these experiments over here that they actually go after a proper target. So all of them are killing. And for this, we made knockouts. So you see this is a Noun6 that contains both, either one of them or nothing. That's the control. And you see the lyses, the specific lyses, not in a control, and with all 3, meaning that all of them are actually working. All right, so that's the efficacy part. Few words about pharmacokinetics, really focusing on the biodistribution, actually. I know it's a big table and a rather busy slide, but what I wanted to say with this slide is that when you do a biodistribution, you should follow ICHS-12 for gene therapy vectors, which essentially tells you that you have to monitor. For gene therapy vectors, you have to monitor nucleic acids. You have to monitor the DNA and RNA. What we decided to do is to monitor much more here, because we wanted to know where the LNP is going. We wanted to know whether it crosses to the cytoplasm, so we took an RNA reporter. We also wanted to know whether it crosses to the nucleus, so we took a DNA reporter. Then we wanted to know how it integrates and whether it integrates and how many copies there are per single cell. And then we also wanted to know whether the RNA from the integrated DNA is expressed and whether the protein is expressed. So a lot of different assays that I'll quickly go through. And we also use different timings. So why do we do all of this? So this biodistribution is important because if you think about some of the risks that are associated with in vivo gene therapy or in vivo CAR T gene therapy, they'll be around off-targeting. The on-targeting, to some extent, are also the risk for ex vivo products which are there. But the off-targeting are not. So it is important to see where these particles are going, what kind of potential toxicities they can cause, and also if they integrate in off-target tissue, from a perspective of the long term, what kind of cells are affected, what kind of cells we need to monitor. So a timeline, we inject a particle to mice, 5 hours, we look at the particles labeled with Cy5. We look at the reporter luciferase on RNA, did they cross, are they in the cytoplasm? 21 days later, we look at the DNA reporter, are they in the nucleus? 30 days, we do a battery of tests, including a classical qPCR and RT-qPCR to follow the vector presence, vector integration, and vector expression. And we also look at the protein expression by immunohistochemistry. All right, so let's start with Cy5. What we do here is we have the MC3, which we call Onpattro-like, and we use the untargeted LNPs and targeted LNPs. This is the dose that I showed on the previous slide as being efficacious. We already knew that at this dose, we'll see not much. And that's what you see with this blue color over here. It's too low. So that's why we make the dose higher up in order to visualize where it's going. And the MC3 reference is going to the liver. This is designed for a liver. It should go there. The untargeted LNP, it has this pattern of distribution. This is, by the way, spleen, lungs, liver, kidneys, and heart. With the targeted LNP, you see that it will go to the liver, but already you see a splenic localization. And in the spleen, there are a lot of T cells. Also, in lungs, there are T cells because there's lymphoid tissue around lungs. Now, when we look with messenger RNA, also 5 hours after, so very quick, we wanted to know where this vector actually ended up, whether it ended up in the cytoplasm and where it was. And you can see that, again, for Onpattro, it's liver. For the targeted LNP, the picture starts to get more and more specific. So it starts to go to a T cell rich organ, which is spleen. Then, when we go really 21 days later and look where the vector went to a nucleus, this becomes very specific. So these ones, of course, you don't see them anymore because they are not targeting T cells. And this is targeted LNP that you can see is almost exclusively with this type of method can be visualized in the spleen. And there is something in the lungs as well. So this is just to put it all together on one slide. The particle distribution, where you still see a liver distribution. Then when you go to cytoplasm, it starts to go to spleen. When you actually think about really integration, what kind of cells are affected, it starts to be very, very specific. Then we look with the qPCRs to measure different organs. And you have here the presence of the DNA, so everything that we can measure, the integrated form, which is after DPN1 treatment, and then expression of the C19 CAR mRNA. In general, these signals are very low. And that's signifies here by the limit of detections of the qPCR, which is 83 copies per milligram of DNA, and also RNA. And they are in the spleen. There is something in the lungs, very little in the liver. But still, there seems to be some integrants here in the liver. So we were interested, okay, do we really have some form of the delivery into a liver tissue? And I think in the interest of time, I'll skip this one. And then we wanted to, we were looking for an appropriate method, actually, how to visualize whether this is in the hepatocytes or what kind of cells it is. And all those single cell assays are somewhat difficult because they need to have some amount of cells to detect. And what we are doing here, we're actually looking for a needle in a haystack. But when we started to do an NGS-based method for integration pattern analysis, which is modified LAMP-PCR, we came across a certain opportunity because in these animals that are immunodeficient, those mice, there are two genomes. One of them is coming from a mice, the other one is coming from a human because of the T cells that are injected into those animals. And therefore, if we actually have unique reads connected to the vector, this catalog of all those reads, we can place them either here or here. And actually, if they are placed here, this means it's on target, so it's a T cell, because that's the only possibility in those animals. So that actually allows us to say, I'm analyzing a liver sample, am I a T cell or am I a hepatocyte? If it's a mice genome, it will be a hepatocyte or a Kupffer cells. So what we've done, we took this below limit of detection thing, and we sequence it. In the spleen, you just see the human genome, so it's because there's too many reads. In a liver, you see that 80% of this signal is still coming from a T cell. This is not surprising at the end, because those T cells in those models, this xenogenic model, so those T cells, they are actually going everywhere and reacting to the mice. So they will go into the liver as well. But only 20% of this, which is already almost undetectable, can be associated with a liver mice sample. And then we decided to go even farther to try to see where they actually are. And what we've done here, we connected GFP with the CAR with the T2A system. So essentially what it does is that every expanded T cell that contains CAR and expands because of the reaction towards the antigen will have a marker, which is GFP, because they're on the same genome which is integrated there. And that allows us to visualize where the thing is going. And if you look at the spleen, there is a beautiful homogeneous distribution of T cells. If you look at liver or lungs, you can see that this is some few dots, and they are almost exclusively around vessels. When we got a report from a pathologist, the report said that these ones are mostly Kupffer cells or infiltrating lymphocytes, or T cells that are actually infiltrating there. So again, with all the techniques and assays that we're trying to use, we were unable to visualize in these different methodologies other cells than T cells, which is the target, and those cells that are actually eating particular matter, which is monocytes or Kupffer cells. So that's the biodistribution. Few words, and I'm perfect on time. I have 3 more slides about the distribution patterns. So what you see over here in this analysis, which I already alluded to for a different purpose, we wanted to look at how this transposase distributes the gene of interest within the genome, because that's important from a perspective of safety. And the hypothesis there was, of course, it's a sleeping beauty, it's an enzyme, it has specific activity. It is likely that its distribution will be not impacted by the type of delivery, so we should see similar patterns from what has been published about sleeping beauty. And by the way, sleeping beauty was in a couple of clinical trials with ex vivo CAR T therapies, like for instance Coramba. And what we see here is more or less what sleeping beauty is doing. So it's a random distribution throughout the chromosomes. It is avoiding exons going into introns, and at a certain distance from transcriptional start sites, which is essentially what has been published about the sleeping beauty. Then, one more important safety parameter, which is VCN. VCN is a vector copy number. It has been limited for the ex vivo CAR therapies to be below 5 copies. What we measure in the blood of those animals, it's, I would say, somewhere between 1 to 2 copies per cell. Again, suggesting that what we do is we're hitting a certain small subset of cells that are expanding, and we are taking advantage of the proliferative effect of those T cells. We're going with a very low dose, and this distribution and the vector copy number is limited. So to conclude my talk, in vivo gene therapy for CAR T is really surfacing with all these clinical trials for lentiviruses, for LNP mRNA, as a solution to some of the major issues with scale-up, with the cost, with patient access to cell therapies. What we are trying to do is actually to take advantage of the power of those two modalities, so or some of the attributes. So we want to be integrative. We want to do a single administration at a very low dose, and this very low dose in a combination with amenability of LNP to scale up. And what we've heard today about all the new methodologies and how to actually manufacture and scale up the LNP should result in basically development of a gene therapy vector that would be competitive towards other modalities that you showed during the presentation. What we have demonstrated so far is an efficacy, essentially an expression of a CAR T in T cells, so we can make in vivo CAR T. Those in vivo CAR Ts are functional, they can control the tumor, and they also, importantly, they are long-lasting. So they can persist, they can be evoked. I didn't talk about the phenotype, so we have the data about the phenotype of those cells, and they have a phenotype which is close to a stem cell memory, which is associated for ex vivo CAR T therapies with the good predictions of the outcomes of the clinic. We also are using a very limited dose when you compare to other LNPs are being used, for instance, on Patro. This NCTX display functional very strong tropism towards T cells. So functionally towards the nucleus of the T cells, it's very specific. And last slide, all this work has been prepared by this group of talented young doctors. We have a lot of collaborations with the University of Utrecht, also with Sintef, with Sven. We have also, we've been awarded a nano engine grant from AAC. And I think I'll stop here and happy to take any questions.
Thank you, Dr. Lubelski. Now, let's start with the questions.
Yeah, thank you for this presentation. In your last slide, you write minimal off-targeting. What is acceptable as minimal? Do you have an idea on how much you can allow for with your approach?
So here, the discrimination with the minimal is what you've seen with regards to a delivery of the cargo towards the nucleus of the T cell. Because this is one of the major risks for, let's say, an off-targeting related to insertion mutagenesis. So you'd like to limit this, and by doing so, you'll limit the integration and you'll limit the expression of the protein. To answer your question, what is accepted, I think we'll only know once we'll go through all the preclinical studies and have a dialogue with the regulators.
Yeah, that's exactly my questions. And what would you expect the regulators would like to see for that?
I think that's what's written in S12. So first of all, a biodistribution based on the qPCR to qPCR. These type of studies has been performed for adeno-associated virus, for instance, with regards to integration patterns that has been performed also for lentiviruses with regards to integration patterns. And the vector presence and vector expression, it is also a part of standard GLP tox package.
Okay, thank you.
Welcome.
Usual suspect. So, very, very nice. I have two questions. One related to getting into off-target cells.
Mm-hmm.
So you're injecting IV, you're expecting that if your PK is preferentially going into your T cells, assuming that, the interaction needs to be probably fast. So your targeted LNP need to meet the cell, bind, internalize somehow, or release the payload. But the rest that are do not target for some reason, will be either circulating more or will be eventually picked up in the liver area. I would assume, maybe also in the spleen. These are logical areas. What happens if you get, for some reason, penetration into those off-target cells in the liver and the spleen? What could happen?
So that's exactly the question we're trying to tackle with some of those assays. And what we see is that it will drain to the liver, like everything. Liver is the same, whether you're virus or non-virus, you'll end up there, or some percentage of the particle will go there. We see that it is actively engulfed by macrophages, and that's also is expected. But when it comes to the liver tissue, what happens is that we cannot visualize expression. Even with all the methods, in particular, I push this to the limit with the CAR T2 GFP, T2A GFP, because I wanted to see whether if the CAR is expressed, if there is a proliferation, but I can have GFP signal. We don't see this. Why we don't see this, we don't exactly know, but we also performed a series of experiments in the continuous cell lines. So we took Jurkats, we took HapG2, we took Q7, and for some reason, when it comes to DNA delivery, not RNA, they will get to cytoplasm, but they don't get to the nucleus of those different cells. I don't know. It's a question that we are asking, but essentially there is three logs difference between HapG2 and Jurkats. So you have tremendous, you have a vector that is active in that particular cell type, delivering DNA, and in this hepatic model, or hepatocyte model, it's almost inactive. And we don't know why. It probably has something to do with the biology of a T cell, but not with a single point. I think there are a number of different points that makes this vector working there and not really working in the liver.
So one other question that I have is, what do you think is an expression, DNA expression, that needs to be present in your cells? Can you quantify this? I mean, saying this is expressed, this is not, because in mRNA it's easy.
Yeah.
Right? Predominantly, you need tiny amount of protein to be expressed for vaccines, tiny amount of protein to be expressed for editing, because too much is not good, and anyhow it will not be. So with DNA, this is permanent, right?
Yeah.
So the idea is you get now a permanent expression. How much, or can you quantify how much is fine, how much is dangerous? Yeah, I saw this, but it's not a good answer, to me at least.
Which one?
The slide that you're gonna...
Don't know where I'm going.
Yeah.
So this is a little bit of a measure of expression.
Yeah, this is...
This is MFI.
So...
I see what you mean, but we don't know. What we know is that...
Could it be dangerous that the expression will be permanent, even low amount of expression? I know you're targeting cancer, so that's obvious. The question is, it's not a temporal, as opposed to the other companies that show RNA, it's temporary. They have to do multiple dosing, but
Yeah.
they also have to, they can stop at some point.
That's true, and some people think about switches and stuff like this. But
Exactly.
but
So-
you have risk-benefit profile that comes from ex vivo therapies, which are permanent as well, because they are cells that are permanently modified. They come back to your body, and in principle, they should survive. So the only difference here is that you are targeting these cells not outside in a bag, but you're targeting them in a vein.
Yeah.
So the risk-benefit, I think, from that extrapolation, is likely to be accepted. And also we know that there are lentiviruses, clinical trials, and lentivirus is doing exactly what we are trying to do. They integrate permanently.
But their expression is very low, right?
I think actually our expression is lower because
I'm sure. I'm sure you're lower than
No.
But
so the VCN is important measure here. So the VCN has been, in particular, limited to quantify the amount of copies per cell that you integrate. Because if you have, okay, so the expression, let me go back a little bit. The expression will be driven by two things. One, it will be a promoter. So we have the same promoter as the ex vivo. So the expression based on the promoter will be the same. Then the second one is amount of copies. Yeah. The more copies you have, the more expression, until saturation, of course, you'll get. So the VCN will tell you that you'll limit expression to what has been already observed in a clinical trial and in the products, and has been at least third safe. Right? So that's why you do a VCN to say, I'm below 5, and we are between 1.2 to 2, which is exactly what you want to have.
do you think you'll need to inject another round?
This question we get often. And the ability to re-inject is important, because in principle, when the therapy stops to be effective, and maybe it will happen at certain moments, with these type of vehicles like LNP, you have at least theoretical possibility to redose. But it's a different thing to redose bi-weekly and to redose after half a year, and to redose twice, 3 times, or multiple. Yeah. So if this effect is long-lasting, and you can redose after a year, it might be possible. What worse could happen? You'd create maybe neutralizing antibodies, maybe ADA, but definitely, yeah, it's not a virus.
You're using an antibody as a targeting moiety. Your amount of antibody is very, very minimal compared to protein therapeutics. So the chances that you will get ADA after a single dose exist, but it's limited, I think.
I agree with you. I also think that this is not the biggest risk, and therefore this readministration can be another reason why this type of approach with LNP, and I know I'm preaching to a choir because you are all LNP people, but I think this might be another reason why LNPs will have a certain advantages over viral approaches, over lentivirus. These lentiviruses, when injected once, they've been already associated with some toxicity with the recent publication from EsoBio. What would happen if they are tried to inject it again? They will definitely create neutralizing antibodies. Right. And with this vehicle, it might be different. But time will tell. We need more data. We need more studies to actually look into those things.
Thank you.
Does anybody have a question for Dr. Lubelsky? No? Then thank you.
Thanks very much.